The idea of encapsulating biomolecules in lipid vesicles may sound like a small technical trick, putting proteins or RNA into little bubbles of fat, but in reality, it goes to the very heart of the origin of life and synthetic biology. Lipid vesicles, or liposomes, are simple spherical shells formed when amphiphilic molecules with water-loving heads and water-hating tails self-assemble in water. They look strikingly like cell membranes, and that resemblance is no accident. Life itself is bounded by such membranes. So the question is, if we load these vesicles with the right biomolecules, could we create a minimal form of synthetic life? At first glance, it seems straightforward. If you put DNA and enzymes into a vesicle, the enzymes can transcribe RNA or even synthesize proteins inside. Scientists have already made cell-free systems that, once encapsulated, start producing proteins as though they were inside a living cell. In one famous experiment, vesicles containing RNA polymerase and a template DNA strand managed to generate RNA transcripts, internally turning a bubble into a tiny factory. Isn't it remarkable that such basic ingredients, when sealed in a lipid shell, spontaneously mimic life's processes? But the real challenge and the fascination lies in what's missing. A living cell doesn't just hold molecules. It maintains homeostasis, divides, repairs itself, and evolves. Synthetic vesicles often leak, lose components, or stop functioning after a while. Without nutrient exchange, waste removal, and energy supply, the life flickers out. This reveals a profound truth. A boundary is not enough. For synthetic life, we need dynamic membranes, channels for transport, self-repairing chemistries, and perhaps even primitive metabolism embedded in the vesicle. A lesser-known fact is that vesicles can actually grow and divide spontaneously under certain conditions. If you feed them lipids, their membranes expand until they pinch off into daughter vesicles. Combine this with encapsulated RNA that can replicate, and you begin to glimpse a self-sustaining cycle. Information inside, guiding division, outside. It is exactly this coupling of information, metabolism, and compartmentalization that origin of play researchers believe sparked biology on the early Earth. Could modern labs be replaying that script in miniature? The paradox is that building synthetic life from vesicles blurs the line between alive and not alive. A vesicle with DNA transcription running inside is not conscious, not even a true cell, but it does process energy, copy information, and sometimes divide. Is that enough to qualify as life? Or is life something emergent that appears only when many such systems interact, evolve, and adapt? The haunting insight is that every advance in vesicle-based synthetic biology challenges our definition of life itself. Each encapsulated enzyme, each dividing bubble, each metabolic cycle reconstituted in vitro pushes us closer to an answer. Perhaps one day we won't just simulate life, we'll build new versions of it, starting from lipid vesicles as our synthetic eggs. And when that happens, we may have to ask, did we create life, or did life simply re-emerge from the physics and chemistry that always made it possible? Synthetic life aims to recreate minimal living systems from non-living components. A core approach is encapsulating biomolecules, such as DNA, RNA, and enzymes, inside lipid vesicles that mimic cell membranes. These vesicles provide compartmentalization, separating biochemical reactions from the outside environment. Encapsulation enables control of concentration, protection from degradation, and emergence of system-level behaviors. By reproducing essential functions like metabolism, replication, and signaling, scientists hope to identify the minimal conditions necessary for life. This pursuit tests where chemistry ends and biology begins. DNA and RNA encapsulated in lipid vesicles can replicate under the right conditions. It is like building a tiny house where all the furniture, tools, and energy supplies are contained inside, ready for living. Lipid vesicles are ideal models because they resemble the bilayer membranes of natural cells. Their amphiphilic molecules self-assemble spontaneously in water, forming stable, spherical compartments. Vesicles can be tuned in size, permeability, and composition to control molecular exchange. This ability to mimic selective barriers is essential for maintaining homeostasis. Encapsulation provides a platform where metabolic and genetic subsystems interact without uncontrolled dilution, and such protocells form experimental bridges toward understanding life's origins. Fatty acid vesicles allow nutrients to diffuse in while retaining large biomolecules like enzymes. It is like a coffee filter that lets in hot water but keeps the coffee grounds inside. A key step toward synthetic life is trapping genetic polymers inside vesicles. DNA and RNA serve as carriers of information, enabling heredity and evolution when replication is possible. Vesicles provide a stable environment that prevents nucleic acid degradation by external enzymes. Researchers have shown that RNA polymerases can function inside vesicles, copying RNA strands. This creates the foundation for Darwinian evolution at the molecular scale. Encapsulation thus transforms inert chemistry into systems with memory and adaptability. Vesicles containing RNA replicase enzymes have successfully amplified RNA sequences within their compartments. It is like copying blueprints safely within a locked room instead of leaving them exposed to the weather. Life requires metabolism, not just genetic information. Encapsulating enzymes within vesicles allows controlled chemical transformations to sustain the system. By supplying external substrates, vesicles can perform cascades of reactions inside their walls. These metabolic subsystems can produce energy carriers, such as ATP, or simple precursors for replication. Importantly, vesicle membranes regulate input and output, making the system semi-autonomous. Encapsulation therefore couples information with metabolism in a lifelike framework. Lipid vesicles have been engineered to contain glycolytic enzymes that convert glucose into ATP. It is like stocking a miniature kitchen where ingredients are processed into meals without leaving the house. Beyond metabolism and information storage, vesicles must replicate to qualify as lifelike. Vesicle growth can occur by absorbing additional lipids from the environment, leading to spontaneous division. When genetic material and enzymes are present, this division can propagate functional systems. Researchers are exploring whether vesicle replication can be synchronized with genome replication. Achieving such coupling would allow protocells to inherit both structure and function. This is a critical milestone toward autonomous synthetic life. Fatty acid vesicles grow and divide naturally when fresh lipids are added to the solution. It is like soap bubbles splitting into two while carrying a copy of their contents. Encapsulation also enables information processing, where signals trigger internal responses. Molecules like ribozymes or synthetic circuits can control reaction pathways. This mimics cellular signaling and regulation without full biological complexity. Information processing ensures that protocells can adapt and coordinate functions dynamically. Such behaviors blur the line between chemistry and computation. Ultimately, protocells act not just as reactors, but as decision-making entities. Vesicles with DNA-based circuits can switch internal reactions on or off in response to external cues. It is like a thermostat that senses temperature and decides whether to activate heating. Microfluidics and synthetic chemistry provide precise methods to encapsulate biomolecules. Droplet microfluidics allows production of highly uniform vesicles, each containing controlled molecular cargo. Optical tweezers and fluorescence microscopy enable real-time observation of individual protocells. New chemistries also allow vesicles to be stabilized against leakage and degradation. These tools transform protocell construction from trial and error into programmable engineering. Encapsulation is no longer an art, but an emerging technology. Microfluidic devices can generate millions of identical lipid vesicles, each with a different RNA sequence. It is like a factory assembly line producing identical cars, but customizing the engines inside. Encapsulation experiments reveal how life may have emerged from non-life. They also show how minimal systems can carry out functions once thought exclusive to biology. This has implications for origins of life, research, synthetic biology, and astrobiology. By understanding what is required for lifelike behavior, scientists refine the definition of life itself. Encapsulation thus challenges philosophy as much as chemistry. It reframes life as a continuum, not a binary category. Protocell studies suggest that early life could have started from simple vesicles with primitive metabolism. It is like realizing that the line between a toy wind-up car and a real car is a matter of complexity, not essence. The ultimate goal is to integrate replication, metabolism, and evolution in one vesicle system. Such synthetic life would demonstrate that biology can be built bottom up. It would allow testing of evolutionary principles in controlled laboratory conditions. Applications could include programmable therapeutics, biosensors, and biomanufacturing platforms. Achieving synthetic life would also redefine humanity's relationship with nature by showing that life is reproducible technology. The paradox is that by building life, we learn how fragile and contingent it truly is. A synthetic cell that replicates its genome and membrane could evolve new traits under selection pressure. It is like planting a seed of life in a test tube and watching a new ecosystem emerge.